antibiotics. Okay. So I stepped out, I came back, and all my notes have gone. But I hope you remember what was on the board before. So what I want to do now is actually, tickle your interest by showing you some real data, just to show that it's not abstract. There are simple experiments you can do that push the limits of what is known about biology. So remember I said you grow bacteria in a flask. You can actually shine light through the flask, and that measures the number of scatterers, little discrete objects in there, and that will capture both the number of living cells and the number of dead cells in total. Okay? And that's called OD600. Here I've written a rescaled version of OD600 just to get it into units which are meaningful. You could also do a separate experiment where you measure the number of colony-forming units per ml. It's a complicated name, but roughly all it is it measures the density of living cells. Two different measurements. So I'm showing you some graphs here which actually separately show, in real experiments, the number of total cells and the number of live cells over time. And so here's the first thing you notice. So I plotted log. Okay, this is log as a function of time. And here you see the classic picture, which is pure exponential growth in the absence of antibiotic. Here the antibiotic is called kanamycin. It's an antibiotic that actually binds to ribosomes and irreversibly destroys them, or irreversibly disables them. So this is very nice pure exponential growth within the error bars of the, well, very tiny error bars anyway. This is classic microbiology. And what's happening here is what you all said, that a single cell becomes 2, becomes 4, and so on. And there's very little cell death. So the number of total and the number of live cells pretty much on top of each other. That's why the scale factor is there, by the way. Now, you can ignore the middle part of the graph, but let's just go this other regime where I have a high concentration of antibiotic. Okay? And then as promised, though we haven't run the experiment over several hours, but trust me, if you do, you can extend this curve. You have to work a little harder. What you find is that the number of live cells, which remember is colony-forming units, the number of live cells actually undergoes exponential decay. There's a transient in the beginning. And the reason there's a transient is the way the experiment is done is here, tube has no antibiotic. And at this time point, you dump in that much antibiotic, and then the cells adjust to their new situation, and that's what's going on at this time. But eventually, you get this pure exponential decay. Yes? Right there, t equals 0. So before t equals 0, they were doing exponential growth for enough time. They've adjusted, there's no lag. And at that time, you immediately add the antibiotic. This slope and that one are the same, and then, yeah. So interesting, actually, the number of living cells actually goes up, then it comes down. And in the cartoon, roughly, it means that, you do have a little bit of cell division, but eventually, most of the cells die. Yes? These two? This is not log. This is linear. This is the whole point. I'll get to that in just a second. So what I want you to notice here, unlike up there, which you can't see anymore, but those two curves were two perfectly straight lines all the way through. Living and total were the same, up there. But here, just forget the transient. What happens is eventually, the number of living cells is falling exponentially. The number of total cells is flat, and this makes sense. Because once all the cells are dead, the total number can't change. Right? And so that's your flat point. Now, as promised, there is some sort of boundary between these two, where the living cells here are going up exponentially or going down exponentially, where the number of living cells is flat. Not the total. The number of living cells is flat. And at that very finely controlled antibiotic concentration, which is called the minimal inhibitory concentration, on a log plot, the total number of cells does something, it's not quite exponential growth. Actually, it turns out to be a logarithmic, because on a linear plot, that thing is a straight line. Okay? This also makes sense. What's happening here is that the number of living cells is flat. Remember I asked you what happens at that axis. This is that when the antibiotic curve hits the axis, this is what's happening. The number of living cells is staying constant. But the system is still dynamic. Because obviously the total number of cells is still going up. Now how can that happen? The answer is actually quite simple. It happens because of this. Roughly, every cell when it divides, on average, one of its daughters survives and the other one dies. And therefore, the division and the death rates, as somebody mentioned, are equally balanced. That's by definition, because we work very hard to get that balance point. Right? And when they're equally balanced, the number of living cells cannot increase. But the number of dead cells obviously increases in proportion to the number of living cells. And so the total number of cells, living plus dead, just increases linearly. Okay, is everybody happy with this? This is the phenomenology. This is what one is trying to explain. And everything I'm going to talk about now is just ways to unpack these results. So to summarize, for 0 or low antibiotic, living in total, both exponential growth. For high antibiotic, living in is exponential decay, total is flat. And for a very finely tuned antibiotic concentration, living is flat, total is linearly increasing, not exponential. Total cell is OD600. It's actually 8 times 10 to the 7 times OD600. It is continuous, and I'll show you more of that later, right? But let's now start to understand. So now we just want to capture the phenomenology of it, right? So this is too far, right? Is this okay? For people over there, can you see here? Okay. So we want to write down 2 sets of equations for the number of living cells and the number of dead cells, where the total number is CL plus CD, which can also change over time. Okay. So we have to make some sort of assumption, approximation. We have to have some model. So for the moment, we're going to make a very silly model, where we model each cell as a fundamental unit that can undergo independent death or decay in the same way we looked at in the first half of the lecture, which is we assume there's a constant probability per unit time that any cell divides or dies. Keeping in mind that this cannot be the true story. It doesn't make any sense based on how we know cells are made, okay? But anyway, let's see how far this goes. So I'm going to invent a division rate, Phi plus, and the total rate of creation of living cells is proportional to the number of existing living cells. This on its own would give you exponential growth. Right? But you also have a death rate. Okay? And the total number of dead cells comes from that. Yeah? Very, very simple stuff. So roughly what these 3 regimes correspond to is up here, the death rate is 0. The number of living cells grows exponentially, and the total number of cells is the number of living cells, and therefore also grows exponentially. Down here, Phi plus is 0, Phi minus is very high. Okay? The number of living cells falls exponentially. The number of dead cells is just living cells converted to dead cells, so the total number of cells doesn't change. Okay? And on the axis, these 2 are equal, and if you just plug that in, of course, the number of living cells is constant, and the number of dead cells is increasing at this rate. And therefore, the total number of cells is increasing at this rate. So to put it bluntly, the slope of that curve divided by the number of cells represented here is the death rate per cell. Okay? The slope of that curve is the division rate per cell. And if you just plug in the numbers here, you can work out, suppose this rate in some normalized unit is 1, right? The death rate here is actually 60% of the division rate here. Therefore, the division rate here is also 60% of the division rate. If we can calculate phi minus, and we know phi plus is equal to phi minus, we've also calculated phi plus. Okay, so I've just told you something rather interesting. If there's some rate of division here at zero antibiotic, the rate of division here at fairly high antibiotic is almost 60% of the antibiotic-free division rate. So cells are still growing, roughly as rapidly as they were when there was no antibiotic. What's the difference? They're also dying. Because they're dying, the number of living cells stays constant. So this is, but of course, these equations cannot possibly give you curves of this type with some transients and all kinds of interesting behavior, right? So what we're going to do is to derive a set of curves that are able to do that. And just to give you a sneak preview before I close this up and move on to somebody asked, if you do an antibiotic series, what do you get? This is the actual experiment. Okay, these are the experiments. This is OD, this is CFU. It's the same data that I showed you in the previous slide, but it's just rearranged in some way. Time, log of OD, time, log of CFU. These are total cells, these are living cells. And you can see that you go from exponential to saturating. Here you go from exponential growth to exponential decay via that point. And we're going to make a model whose predictions, with basically one free parameter, captures all the qualitative behavior you see over there, including this little bump, right? Including the very rapid transition from growth to death at very small changes of antibiotic concentration, and various other nice qualitative features. Okay, so the question is, what is this model? How complicated is it? How do we get these curves from such a model? And what can we learn about the cell based on this? Let's start to unpack it. The first insight that one gets is what I had said just before we broke for coffee. In a population, and in fact, you'll see repeated examples of this through the school, when you're going to hear about host-pathogen interactions, or in fact, gene dynamics and so on, you find situations where macroscopically observable things, in this case, things like OD and CFU and so on, things that you don't need a microscope, you can just do the experiment and measure, don't make sense unless microscopically things are stochastic. So there's sort of a trend these days to prove something is stochastic by going all the way down and looking at a single molecule and saying, there, it's stochastic. Right? But for example, statistical mechanics was not built like that. Statistical mechanics was built by looking at pressure and temperature and things that are macroscopically observable and inferring that there must be some underlying noise. Right? And in biology, you can do the same trick. There are cases where phenomenology, macroscopically observable, is a strong hint that microscopically something is being very noisy, random, stochastic. In particular, the fact that cells decay exponentially, as I already said, means that some cells die early and some cells die later. And automatically, that must mean that there's something different about these cells. They're not all the same. There is variability at a single cell level. You infer that. Now, the model I'm making just takes that variability seriously and tries to describe it in literally the simplest possible way. If that simplest possible way doesn't fit the data, we're going to make it more complicated and so on. Okay? Is everybody clear what the way we're going to approach this problem is? It's not to say that the model I'm going to make here is the truth. It's just the simplest possible model that we're going to compare with data and then add more bells and whistles as required. So let me unpack this. So we know that this equation is not correct because cells are not fundamental particles that have constant probability of death and division. In fact, we automatically know this equation is not by comparing with the data because this equation will just predict straight curves. It cannot predict any transients. The fact that you have transients roughly means something like these rates are time dependent. And if you're going to add time-dependent rates, there's no point writing an equation like this since I haven't given you a recipe for how to calculate these time-dependent rates. Okay? So let's now imagine that a cell is not an independent object, but it consists of some microscopic things. and I don't know what they are, so I give them a nice name. They're called widgets. A widget is just a fanciful name for a little object that you don't know what it is. Not a real molecule. Okay. So, the widget has the property that it is the fundamental unit of replication in the cell, in my model. That means one of the things the widget does is if you give me a single widget and you wait a little while, you can make 2 widgets. And you need this widget to make those. Okay? So the number of widgets increases by 1. This is going to be an ingredient of a master equation, which was over there, but now it's erased. We're going to put it back. There's another thing widgets can do, which is that they can get destroyed. This is an example. If I add an antibiotic and it destroys a ribosome and then destroys the collection of molecules around there that are responsible for self-replication, you destroy the widget. Therefore, the number of widgets goes down by 1. And I'm going to put rate constants now, not rates. Just like we had RNA decay with a rate constant gamma, so the RNA equation was alpha minus gamma x. In this case, the degradation is a rate constant, but so is the synthesis. So now our equation for widgets, so this is for cells. Our equation for widgets, put that in quotations because you shouldn't take it too seriously, is the following thing. Unlike the mRNA case, where this was just alpha, now it's alpha x, because each widget is the substrate to make more widgets. It's not a fixed gene that's making RNA. And if you want to focus on something concrete in your mind, you can imagine widgets such as ribosomes. Ribosomes spend all their time making more ribosomes and spend a little bit of their time making all the other proteins in the cell. And in fact, that balance, you can read all of Sanjay's papers on this particular choice of ribosomes making ribosomes versus ribosomes making everything else. Anyway, now simple model, you get...
Sorry, which particular paper? The one you sent?
There's a recent one with Parth, and this is published where? I can share. So the reading has been given. I sent a manuscript with that reference is in there. Okay, it'll be Jane and Pratim. Yeah, Parth. Yeah. Okay, it's in the references. One of the first few references. Okay. Reference 10. Okay, thank you. So this is a slightly different equation than the one we had earlier, right? Only slightly, only slightly. But the implications are sort of enormous, right? We'll see what that means. So roughly you get an equation that dx dt is alpha minus gamma x. This is just for widgets, right? And the solution to that equation is well known. It's e to the alpha minus gamma t. Okay, this is microscopic. And what I'm saying is it's reasonable to assume that at some level of coarse graining inside a cell, we know a cell is not a fundamental object, but somewhere in the cell we can imagine there are fundamental objects that do behave like indivisible particles that you can destroy or create or self-replicate. But how do you go from that to cells? You have to make some further assumptions, and here's my assumption. So this is the most important part of this whole slide. I imagine that a cell is a bag of widgets. A bag of widgets. And widgets can replicate, or they can be destroyed. What I say is that when a cell hits a threshold number of widgets, in this case 5, omega equals 5. I'm just making this up. When a cell hits a threshold number of widgets, the cell divides. And when the cell divides, the widgets are shared with equal probability between the two daughters. It doesn't mean exactly 50-50 because that itself is a stochastic process. So you get division. There's something else that happens. Suppose you lose a widget, and finally lose your last widget. If you don't have, for example, ribosomes, you can never make more, ever. So that cell is dead. And this is quite a reasonable thing, right? Once you lose your last, most basic self-replicating unit, you can't make any more cells. Yes, question? Oh, no, the threshold is arbitrary. This omega is the free parameter I had described earlier. Okay? Yeah. Okay. So just to be concrete, remember, if you increase the antibiotics, something roughly, if you increase antibiotic levels, roughly alpha decreases and gamma increases, but you can imagine various other ways. In this case, alpha is constant, gamma is increasing. Okay. But the cool thing is the following. You've now gone from a single particle type of model to a bag containing a bunch of particles. And if you zoom back on this process, just zoom back, zoom back, zoom back, and shield your eyes to the fact that cells are bags of particles, it starts to look again like this process, where you have a cell which can replicate, like so, or you have a cell which can die, like so. And we had made this totally arbitrarily parameterized, obviously wrong, effective parameters for the rate at which cells duplicate and the rate at which cells decay. Now what I'm telling you is, you can calculate these effective parameters from those parameters via this recipe. And if it looks complicated, it's not that complicated now that I've taught you how to do master equations. Okay? So let's see how to do that. See how to. So this again is just like I had drawn here, and it's gone now, but I have bins. And previously there were bins of how many mRNAs there were in a cell. Now they are just bins of how many widgets there are in a cell. Okay? And instead of P0, P1, P2, P3, I call it C0, C1, C2, whatever. The number of cells with that many widgets. W, W minus 1, W plus 1, and so on. In the same way that cells can hop from one bin to another because of mRNA creation or mRNA decay, now cells can hop from one bin to the next because of widget creation and widget decay. And all we do is include that equation's parameterization in here. So the rate at which, so there I've written it as x, but the rate at which you jump to the right is the number of cells you had here, multiplied by the probability per unit time of a single cell jumping to the right, which according to that equation is alpha x, or alpha w in this case. W is an integer. Okay. And that's alpha 1, alpha times 2, alpha times 3, and so on and so on and so on and so on. Yes? W is the label of this bin. And this bin is, say, bin where W equals 5. This is the total number of cells that have exactly 5 widgets. Again, that funny thing where now the state variable is the height of each of these bins. The number of widgets is just the label on the x-axis. Okay? Yes? Yeah, so this thing is the same as mRNA. Remember, for mRNA, we had alpha minus gamma n. Right? So the rate of creation was just alpha times the number of cells in that bin. In this case, the rate of creation is alpha times the number of widgets times the number of cells, because of this funny property that widgets make more of themselves. It's different than RNA. But the reverse process is the same as for RNA. Right? We had gamma times n times Pn. Here we have gamma times W times CW. Okay? That's the rate at which individual cells transition from a bin to its left. So far, these arrows and these arrows are exactly what I had taught you and drawn earlier. Now we get two interesting things happen. In the mRNA case, even if you had 0 RNA to start with, you could still create more because the DNA was the substrate and that wasn't going anywhere. But now, because widgets are a substrate for their own creation, once you get to 0 widgets, alpha times 0 is 0, so you don't get an arrow here. That corresponds to the idea that once you've got a dead cell, it can never go back. Right? That's the missing arrow. Do you see that? That's the left side. You can lose your last widget, but you can never come back. Now, more interestingly, you have this side. And so this I should really unpack and explain in quite a bit more detail. Here you have a cell, or here you have the total number of cells with omega minus 1 widgets. In this case, omega is 10. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here you have the total number of cells with 9 widgets. And our rule is that as soon as a cell gets 10 widgets, it divides. That was the rule in this cartoon. Now, how do you model that? It looks rather different than the kind of framework I told you earlier, but it's not that different. What happens is, once any cell here jumps to the right, what's the rate at which that happens? It's alpha times W times the C. So in this case, it's alpha times omega minus 1 times C of omega minus 1. This is the same rate as that. Just for W equals omega minus 1, right? But what happens to this cell? As soon as it's got omega widgets, it immediately divides. And if it immediately divides, what's the product of that? It's two cells. And what two cells are they? For example, it could go here and there, right? Or they could both end up here. But the only constraint is that the sum of these two has to be equal to omega. Okay? And each one of these is binomially distributed because each widget has an equal chance of splitting between the two daughter cells. That's an assumption. So now I want you to step back. Just put all this in a black box and just look at the two boundaries. What do you see? What you're seeing is that there is a distribution of widget numbers per cell. Not all cells are the same. And if you just focus on that side, there's some rate at which the cells will move out and die. And that rate is simply the rate of this arrow. So it's gamma times C1, because W equals 1. Yes?
Is it going to be stochastically the method that you choose which, how many widgets are in the divided cells?
Yeah. So that you choose by a binomial distribution. Stochastically choose that. You'd use that.
How is it distributed?
Oh, so each widget, there are 10 widgets, right? Each widget has a 50% chance of being left or right. So the number of widgets on each side just follows a binomial distribution with parameter 0.5 and n equals 10. Okay, that's what this is. Omega choose W. This is the binomial coefficient. Yeah. Cells as bags of molecules. Yes. They're not in this model. There is no messenger RNA in this model. These are just widgets. Yeah. Remember what I said. I'm not saying this model is the truth. I'm saying what is the simplest model I can make that can replicate the experimental data. We can discuss philosophy of science of whether this proves anything about the model later. This is not a model of, I'm just making an analogy between the way we did the mRNA calculation and the way we're doing the widget calculation. It's the same mathematics, that's all. Okay, I'm not talking about genes, transcription, translation, nothing. Omega is a fixed parameter. You decide. You provide it. Later we're going to see what happens as you vary omega, alpha, gamma, all these things. Okay? But roughly, let me explain what's going on. There is a distribution, right? So remember, already I said, if you put all this in a black box, put all the gray, well, put it in a gray box, right? Put all these in a gray box. That's the total number of living cells. The total area of the gray guys is the total number of living cells. Which is what I've written there. CL is the sum from W equals 1 to omega minus 1 of CW. This red bin is the total number of dead cells, which can only increase, it can't decrease. Cells exit to the left and die. When they exit to the right, what happens? When a single cell exits to the right, it becomes two cells. Like so. And those two cells drop back into two different bins. Which bins they are chosen at random. Okay? Therefore, the rate at which you cross the right boundary is the rate of division. The rate at which you cross the left boundary is the rate of death in this simple model. And if I'm able to calculate the shape of this distribution, somehow, and therefore infer the rate of this arrow, because remember, this arrow depends on the height of this bin. It doesn't just depend on the fundamental parameters, it depends on some dynamical property. And because this dynamical property is changing over time, because the number of cells in each bin is changing over time, those phis are time dependent. The effective phis are time dependent. Okay? So keep that in mind. So what I eventually want to do is calculate the shape of this bin, the chance of the rate at which you exit to the right, and the rate at which you exit to the left. So let's start to get more detail here. Since this is a tutorial, I'm really going to make you now work hard to write down the equation that corresponds to this. Is that fine? That's the whole point of this, right? To get you to write down an equation, and this is just to incentivize you that this kind of hard work is useful. Let me erase this, and hope I don't make a mistake here. Or if I do, I hope you're going to correct me, okay? So what is the kind of thing we want to write down? We want to write down dCW dt. Okay? So dCW dt is that guy minus that rate and minus that rate. Minus the rates of exit, so you get minus CW times, you get alpha W plus gamma W. Right? The first alpha W is that arrow, the gamma W is that arrow. You also get plus. You get cells that are being created from the left. Alpha W minus CW minus 1 times alpha times W minus 1. That's the label on that arrow, though it's not drawn in this diagram. And plus CW plus 1 alpha W plus 1, that is this arrow. Gamma. Thank you. See? Just testing you. So this is good, right? This is a simple master equation like we did before, and it's a nice coupled one-step master equation. It's called one step because each bin depends only on its left and its right bin. But, okay, good. So let's assume for the moment that CW for W less than 0 is exactly equal to 0. Okay, we just identified that, right? Because there's no way to actually transition to something with minus 1 in any case, because there's no arrow that goes here. Okay, so we just write this, and then this is correct, right? Now, that's the left boundary, it's taken care of, but the right boundary is not yet taken care of. Okay? We also write down C of omega is identically 0. There's nothing in the omega bin. Nothing there. Because as soon as you get to that bin, you divide. But this is a problem. Because that means that the number of cells with, let's say, 3 or 4 or 5 widgets can also increase because a cell dropped in from there. So how do you write that down here? You have to write down the rate of these arrows. And in fact, those arrows will feed into every one of these little bins, right? So now, if you guys are following me tightly, you should be able to tell me what term is missing in this equation. How do you write that down? How do you write down the last term? Okay, if I write here, can everybody see? Okay, so there's a plus, right? There's a plus because there are other ways for the cells with that many widgets to emerge, which is dividing of a parent cell. Now, what is the coefficient? So first of all, the coefficient of that must be the rate of that arrow. Okay, so let's write that down. So it's alpha omega minus 1. Now, it's going too far to the right, so I'm worried that you may not be able to see. So I'm going to write it here. There's a plus, okay? Alpha omega minus 1, C omega minus 1. But that flux gets divided into all these bins. And so the flux going to any given bin must depend on this. Yeah? Now, this is binomial distribution, but there's something a little bit wrong with it. This is your traditional binomial distribution. And it's normalized, assuming that cells can split up anywhere from 0 to omega. Okay? But remember, as soon as a cell has omega, it'll immediately divide again. So if a cell actually divides in such a way that, again, you get 0 and omega, that actually hasn't divided at all in our way of thinking, right? Instantaneously keep dividing. The simplest way to deal with this mathematically is just to say that the 0 and omega bins don't exist. So in fact, the normalizing factor here is wrongly written. Normalizing factor is actually 1 over 2 to the omega minus 2, because the left and the right bins, the binomial distribution are actually 0. Right? And of course you get your omega choose W. Okay? And by the way, you get a factor of 2 here, because there's 2 ways of getting something. No. On the line. Because the 0 bin and the omega bin are removed from the normalization. So the sum of all the binomial coefficients is 2 to the omega. The sum of all the binomial coefficients leaving the left and the rightmost bin is that minus 2. Okay? This factor of 2 is simply because there's 2 different ways. If there's 10 widgets, you can get 3 and 7, or you can get 7 and 3. Okay? What I'm telling you all this for is, when you write down a model like this, and then translate it mathematically, then you have to do a little bit of thinking to make sure that you've represented everything correctly. And if you make mistakes, and I did when I first started on this normalizing factor or this factor and so on, when you do the computer simulation, it comes out nothing like the analytic calculation. And that's a very good way to check what you're doing. No matter how smart you are, you're going to be missing factors of 2 and perhaps minus signs. Right? So do the simulation, and it's true. It's probably, when you write down a simulation and analytics, you're going to make mistakes in both, and that's fine. Right? So eventually you have to make them both come out to be the same. Right? So when you're doing stochastic processes, it's terribly easy to make mistakes of this type. Anyway, now we're done. Question? No. This is 2 to the normalizing factor for the binomial distribution is 2 to the n, big N, not 2 to the k. Because sum of the binomial coefficients from k equals 0 to n is equal to 2 to the n. Yeah. Yeah, but then you can do that by just adding another bin and calling it omega plus 1. There are many ways to do this, right? So now you're talking about a slightly more complicated model where you have a transient and there are sort of cells that get nothing, and there are many ways to do this. That seems simpler to you. This is nothing. Doesn't do anything at all. Okay? Fine. Now, let's just stare at this for a second. This is not a master equation. Okay? In the following sense. Remember, the master equation was an equation for a normalized probability distribution. The probability distribution's area does not change over time. Right? Here, we are explicitly considering cells that jump one side to the other and so on and so forth. But what looks like a master equation is actually simply an equation for the population distribution of cells over various bins, but the number of cells is not constant. Okay? So for those people who are following technically, you cannot use this to generate the stochastic process of a single cell. We will get to that in a second to answer a very relevant question, but this, not that. Let's do that. Let's do that. So he said all the terms are still linear. You can still divide by n. This will still work out. So let's do that for a second. Okay? So CL, which is the number of living cells, is the sum from W equals 1 to omega minus 1 of CW. Okay? And I could normalize everything by the number of living cells. This is an example. So let's do that. Before we do, this equation, it looks too complicated and it's messing with your mind, right? It's actually not that complicated. The right way to write this equation is rather simple. You make a vector C, you call CW a vector. C till C twiddle is a vector whose components are C1, C2, C3, C omega minus 1. And then the entire equation just becomes the following. In this case, it's a linear equation. Therefore, you can write it as a matrix. It's just a way of bookkeeping. And in fact, unlike the mRNA problem where the matrix would have had infinitely many rows and columns, in this case, the matrix has a finite number of rows and columns. And so, in fact, there's nothing that stops you from literally writing this down. You can take a full page of your notebook and just write the whole thing. Okay? Now, for a master equation, the total probability cannot change. This equation, the total number of cells can change. And so what you find is that the largest eigenvalue of A is not 1, like it should be for a Markov process, but it's actually greater than 1 or less than 1 or so 0, sorry. It's not 0, but it could be greater than 0 or less than 0. Okay? Now what happens here is there is an eigenvector, and let's call the eigenvector F. Right? So by definition, eigenvector has the property that A times F is equal to some lambda times F. Yeah? Because total number of cells is not conserved, lambda can be greater than 0, in which case the total number of cells is increasing, less than 0, in which case the total number of cells is decreasing, or exactly 0, in which case the total number of cells is fixed. Number of living cells, right? Because this is going from 1 to omega minus 1. So I can actually decompose, oops, loading photos, okay, that's not good. I can actually decompose this, oops. I can decompose this distribution into two very natural objects. One is a normalized distribution of some shape, and the other is the area of the distribution. Okay, and when I do that, it then becomes obvious that the equation for CL, once the distribution has reached its steady state shape, then the equation for CL will look like so. dCl dt is lambda times CL. That's just restating this. Yeah, question?
Just from your model, I'm just trying to connect it to the earlier classes so that I can understand whether I am understanding it or not. First, there are two pictures, the widget pictures and the cellular picture. So may I first understand that omega, or this is a particular type of bacteria, am I right?
Yes.
So their fitness is inversely proportional to omega, or fitness decreases with increasing omega?
I haven't said anything like that. When a cell hits omega widgets, it divides. I'm not talking about fitness in the evolutionary sense. Nothing like that.
Okay. Okay. And the second thing is that given an antibiotic strength,
Yes.
means gamma.
We'll get to that in a second. You're asking how this relates to antibiotics. I'll get to that in a second. What is the physical meaning of all these parameters, the biological, biochemical meaning? I'll get to that. Thank you for that question. Okay. So, where was I? Fine. So what I've tried to explain is, you can actually very easily, if you have a computer, feed in, let's say omega is equal to 10. If omega is equal to 10, with about 5 or 6 minutes of work, you could feed in each of these coefficients of that matrix, each term of the matrix, and you could feed it into the computer. And then for given values of alpha, gamma, and omega, you can ask the computer, what is the eigenvalue? The eigenvalue would be either positive or negative, the largest eigenvalue. And you can say, what is the eigenvector? The eigenvector is the shape of this distribution. And once you know the shape of the distribution, you can actually calculate the rate at which you go off to the right and to the left. The rate at which you go off to the right and the left will be proportional to the total cell number. If you normalize it by the total, if you divide this by CL, then that precisely is the per cell division rate. That is the effective per cell division rate. And this is the effective per cell death rate. So the way to calculate phi plus and phi minus is reasonably complicated. You open up your MATLAB or R or whatever your favorite environment is, you feed in a matrix with all these terms. Once you've fed in that matrix, you say, what is the eigenvector with the largest eigenvalue? Boom. Once you get that, you then calculate the height of this bin and therefore the rate of the right and left transitions. And therefore you get, this is important, phi plus as a function of alpha, gamma, and omega. And you get phi minus as a function of alpha, gamma, and omega. Through this complicated, long-winded process, which is basically solving that cartoon. Okay? So now we've done something quite beautiful. We've taken what used to be sort of molecular parameters, which are alpha, gamma, and some sort of division parameter, omega, right? We've taken a basically molecular model, and we've used it to derive effective parameters at the level of the whole cell. And although this model is far too simple to have any claim to be a real description of a cell, this process is actually totally general. No matter how complicated I make my composition space, my chemical space, it's not one widget, but it's 5,000 different genes and each of their concentrations and all kinds of things. Eventually, the distribution of cells over composition space reaches a steady state. Once that happens, you can calculate the rate of division and the rate of death according to whatever law you want to give for how those things work. And once you do that, you can calculate the effective rate of division and death per cell. I call it the effective rate because you blind yourself to the idea that there are variations between the cells. You just divide by the total number. But you suddenly see that if you start off with all cells with exactly this many widgets, this shape must change over time before it reaches a steady state. That means phi plus and phi minus must change over time. That's precisely why, when we compare this, sorry for these long sojourns, it's not PowerPoint, so I want to make sure This behavior here, in the beginning, is precisely cells growing in the absence of antibiotic, and they reach a certain distribution shape over widgets. Then you add the antibiotic immediately. The distribution then relaxes and moves towards the new eigenvector. And while that's going on, it takes time, and that's what's going on here. Once you hit the new eigenvector, everything becomes e to the lambda t, where lambda could be positive or negative. Okay? So that's one thing I want to explain. There's one other thing which is kind of cute. I leave it to you to calculate over lunch. You can make this matrix, and you can, in fact, simply calculate its largest eigenvalue. Okay? I'll tell you what it is. The largest eigenvalue of this matrix is alpha minus gamma. Don't take my word for it, go and calculate it. Okay? So now we have this rather remarkable equation. I erased certain equations earlier, but remember, dCl dt, effective parameters, was written as phi plus minus phi minus Cl. The rate of change of living cells is the rate of division minus the rate of death, total thing times the number of living cells. We know from this analysis it's also equal to lambda times Cl. Know what lambda is. So I'm going to write down a rather remarkable equation here. So it's not a free-for-all. Turns out that the rate of see if this works. Amazing. Technology. Okay, so it turns out that the effective rate of division minus the effective rate of death per cell, which is a cellular level property that you derive through all this circus, right, must be equal to the rate constant of widget replication minus the rate constant of widget decay. But it's not that phi plus is equal to alpha and phi minus equals gamma. It's not the case. So there is some amount of constraint, but there's still some amount of flexibility. And to really sharpen this question, let's move right down to the point where there is only one free parameter left in the system. If I work at that special antibiotic concentration where phi plus and phi minus are equal, right? Alpha and gamma are equal, it still leaves the open question of what exactly is that phi plus and phi minus value. Is that clear? They could both be 0 because alpha is equal to gamma. So the right side is 0, for example. This is a so-called minimal inhibitory concentration of the antibiotic, right? They could both be 0, and that's perfectly fine. They could both be incredibly high, and that's perfectly fine. So which one of it is going to be? Okay. So this is where we now come back to the idea of stochastic processes. And for that, I want you to zoom in on this graph. Please. F is the eigenvector. Phi plus and phi minus are the per cell division rates. It is rates. Yes, so once you've reached a steady state of F, so this, yeah, so in some sense this is only for large, yeah, so large time, thank you. So as t goes to infinity, thank you. Otherwise phi plus and phi minus will be time dependent and there's no reason for them to satisfy this condition. Okay. What is now happening? Now that you have a model, you can start to unpack further details of the model. And again, philosophically, it doesn't, this whole exercise is not meant to prove the model is right and you shut shop. It is that once the model starts to give reasonable looking things without much work. I mean, this is literally the simplest stochastic model of cell division and death you could make. This is not bad. So now you want to understand why it's doing that. And let me explain. When a cell divides, the immediate post-daughter cell in this model has a distribution of widget numbers, which in this model is binomial. In a more complicated model, it could be more complicated. It doesn't matter. Correlations between the two daughters. I mean, there's lots of things, right? But still, there is an underlying distribution of widget number in new cells. And depending on how many widgets you happen to inherit, you can ask, what is the chance that you make it to the right before you get killed? Okay, I'll tell you what those are. So the only curve I want you to focus on now is this gray thing, which is a traditional histogram, which is just a binomial distribution. That says what the initial state of the cell will be immediately after division. Remember, I drew a diagram of an immediate post-division cell. That's the state of the immediate post-division cell. Now I ask, what is the probability that that cell will divide rather than die? That's what these curves are. These curves are those quantities for different concentrations of the antibiotic. In other words, for different values of gamma at fixed alpha. So this is gamma over alpha is quarter, half, 3/4, 1, 1 1/3, 2 and 4. Right? So more and more antibiotic. As you have more antibiotic, it's only cells that accidentally already inherit large number of widgets that manage to divide. All the others die. Very low antibiotic, almost all the cells divide. Very few of them die. My question to you is, this is a pedagogical class, how did I calculate these curves? Okay. So this is the last thing I want to talk about because I want to leave 20 minutes for questions at the end. So how did I calculate these particular curves? Okay. And this is something called a first passage time problem. What is the chance that I hit the right boundary before I hit the left boundary in some space? And you can see that it's a very clear physical thing, but you're dividing and dying, right? So these are very natural questions to ask. And stochastic processes framework allows you to find very beautiful answers to those questions. Yes? That's a first passage time problem. Yeah? So you're going to see many first passage time problems. So when you have my standard picture of a stochastic process is wiggly curve, what is the chance that you hit a boundary? Or what is the chance that you hit one boundary before hitting another boundary? In this case, this is division and death. That's the number of widgets. And single cells are bouncing around. And if they happen to hit omega in this model, they'll divide. But if they happen to lose the last widget, they will die. And these are the kinds of things you could measure in an experiment. In fact, Shashi is doing these experiments. Okay. Now, before we finish and start to do this calculation, I'm going to put the screen up and so on. I just want you to come back to this question, which biologically, the first passage time problem is biologically, it's nice, and it's nice to do the analysis, and I'm teaching you how to do it. The biology question here, more interestingly, remember I said, if alpha equals gamma at that precise antibiotic concentration, phi plus and phi minus have to be equal, but how big are they? And that's the last thing I want to show you before going to this first passage time calculation. Good. This actually fills the screen nicely. And somebody else asked, what are phi plus and phi minus as a function of alpha, gamma, and omega? What actually are these functions? Here, I'm showing you that entire thing. Okay, so let me explain how it's done. These are all histograms for omega equals 10, a low number of widgets. These are all histograms for omega equals 50, a high number of widgets. Therefore, there'll be 0 to 10 and 9 non-zero bins, or 0 to 50 and 49 non-zero bins here. Okay. These are the steady state shapes shown at fixed height. Okay. Therefore, the rate of exit to the right is proportional to the height of the bins on the right for different values of the antibiotic. And those values are shown here, 0, 0.51, 1.52, 2.5. And here you can choose your units of time to be 1 over alpha, in which case the only dimensionless parameter that's relevant is gamma over alpha. Okay? And obviously then gamma over alpha equals 1 means alpha equals gamma, means you're at the minimal inhibitory concentration. That's the very important. At this concentration, phi plus and phi minus are equal. But, strangely, for low omega, phi plus and phi minus are both quite high. For high omega, phi plus and phi minus are both about 0. Okay? So, roughly, phi plus decreases as you increase gamma, phi minus increases as you increase gamma, as you would expect, but in very different ways depending on the value of omega. And now that you've got that idea, I'm just going to plot the whole result on this extremely complicated picture, but I'm going to walk you through it. Yes? Yeah. Pretty much here, phi minus is very small. Yeah. So in fact, at gamma equals 0, phi minus is 0 because you can't exit from the. you never lose widgets. Okay. So in fact, phi minus is 0 phi minus is identically 0 for gamma is equal to 0 for any value of omega. No, I'll show you that. That's what these curves are. If I were to plot the magnitude of these arrows as a function of gamma of alpha, which is plotted here, omega equals 10, that's this curve. It's the dark brown curve. Okay? So phi minus starts off at 0, then becomes larger and larger and larger and larger, like that. Phi plus starts off at 1 in natural units and drops down like that. And phi plus and phi minus are both equal when gamma over alpha is equal to 1. Okay? This is for gamma is equal to 10. This guy is for gamma equals 50. Oh, sorry, omega equals 50. Thank you. And this is the last thing I want you to track. Look at the darkest curves here. They are actually extremely simple. In this situation, large omega, as you increase the antibiotic, the division rate drops to 0 and then stays at 0. So with less than minimal inhibitory antibiotic, cells divide, but they divide more and more and more slowly. Until you get to this point, in which case they don't divide at all. Correspondingly, cells don't die at all for low antibiotic concentrations. But once you cross MIC, minimal inhibitory concentration, their death rate increases even though their division rate is 0. This is the picture you get in textbooks. In a textbook, below inhibitory antibiotic concentration, it's assumed that all the cells are dividing, but slowly. Above inhibitory concentration, it's assumed that all cells are dying, not dividing. And at inhibitory concentration, it is not just assumed, but it's enshrined in the word bacteriostatic, that cells are neither dying nor dividing. Which, as I've shown you, is not true. We've measured the division rate at that value, and it's very high. It's nearly as high as without antibiotics. So the textbook picture of antibiotics causing, cell stasis is totally wrong. But it is a limit of the model. What limit is it? It's the limit of large omega. Again, to make it clear, I can plot when alpha is equal to gamma, and therefore phi plus equals phi minus. I can plot whether phi plus and phi minus are both high or both low as a function of omega, and that's what I plotted over here. As omega becomes larger and larger, the phi plus and the phi minus, which are both equal, drop all the way down to 0. Why is omega the relevant parameter? It is roughly the variance of that binomial distribution. It is the fluctuation scale of the Poisson process of creation and destruction. So basically, it's a fluctuation-driven division and death rate that is happening microscopically, even though macroscopically everything looks flat. Okay? So I think this is actually quite a wonderful way to understand what's happening at the single cell level. And it's all inference. We've not actually looked at the single cell level to see whether they're dividing and dying. We're doing those experiments now. And I have strong hope that it will come out exactly this way. Okay, so let's, yes, Patrick. If I can infer it in two different ways. Okay, I can infer omega in two different ways. I can infer omega by trying to do a best fit to these curves. Okay, in which case omega equals 10 is not bad. That's what I've shown here. I can infer omega by the actual measured value of phi plus and phi minus at the minimal inhibitory concentration. In which case it comes out to about 2. Okay? Right. No. Listen. So let me unpack that a bit more. You should not take the model too seriously. There are sources of fluctuations in real cells far beyond the binomial partitioning of widgets at division. In this model, that division threshold automatically controls the noise level. There's no reason for both those to come out of the same process. There could be other additional extrinsic sources of noise. It's not ribosomes. Thank you. So what is the widget? Right now, Jun has done a completely independent type of experiment where they grow bacteria in the presence of different nutrient concentrations, antibiotic levels, and so on. This is also a reference in the paper. And they infer that there's a minimal size of a cell below which it cannot divide using such calculations. And they also get somewhere between 4, 8, 12 units. So that's the number. Okay, and we're getting something like 10, right? So it's not a large number. Ribosomes are there in thousands. So these are not ribosomes. It's a minimal autocatalytic set. Right? So, of which we can discuss what's in it. But it includes DNA replication forks. It of course includes ribosomes. It includes some set of proteins and metabolic net, all kinds of things, right? But the number there are is what we're inferring. And it's a small number. And this agrees with a completely independent way of doing the measurement. It's an interesting question. In fact, I'll take it further. You can actually do an experiment where you push cells to the very limit of depth. Then the composition of the cell is the widget. How do you do that? I don't know. Right? Okay, so can I have this slide up? Is there somebody who can, yeah, and you can turn the projector off? Yes. We're increasing, as antibiotic increases, gamma over alpha increases. Correct. But I don't model that in a detailed way. I just say gamma over alpha increases. The reason I can do that is because I can always go to the point where gamma equals alpha, because I know that's the... So different antibiotics will have different concentrations at which gamma equals alpha. So I don't take antibiotic concentration too seriously. Okay, so let's finish, okay? 5 minutes. How do you calculate this probability of cells? Sorry, I'm just pulling up my notes here so I can make sure I don't make an error. How do you calculate the probability that a cell with a certain initial number of widgets, right, so this is widgets equals 0, widgets equals omega, and I know the initial number of widgets, and this is time. So I start with the cell and I give you the initial number of widgets. And then I allow the stochastic process to continue where the cells will gain or lose widgets. And while you're in this regime, it is basically your original master equation. Because the funny thing that happens on the death side and the funny thing that happens on the division side is not happening by definition. Okay? So in principle, you could imagine, complicated things happening elsewhere. Right? But the idea is, as soon as you hit omega, you say, that's as they say in the TV show. I'm calling it, that's the time of death, or time of division. Yeah? And as soon as it's 0, so that's the time of division, that's the time of death, and I'm done. And so the way I do this, numerically, for example, is I run many different stochastic realizations of the same thing. Right? And every time I run it, I'll get a different time of division, time of death, and this is a bit complicated. These are things that divided rather than die. So my distribution of division times are conditioned on cells having divided in the first place. My distribution of death times are conditioned on cells dying in the first place. And I could also calculate from this initial condition how many of the curves hit that boundary first before this one. Okay? Now, how do you calculate this? Seems very complicated. The way you do it is you take the original stochastic process. Okay? And you say these boundaries are absorbing. Once you hit that, the curve just stops. Because that's what happens. Right? In fact, the left boundary is already absorbing. You never recover from getting 0 widgets. So we've taken care of the left boundary. You take care of the right boundary by just removing this. Once you go off to that side, you don't come back. The cell divides. But you don't care anymore because you're just interested in the time it took to divide first. Everybody with me so far? What I'm saying is reasonably complicated, is usually reserved for the very last part of a stochastic processes class, first passage time problems, and it takes much longer than this to explain in detail, but I'm giving it a shot to try and do it in 5 minutes. I'm actually calculating, I could calculate the first passage time distribution to division conditioned on dividing, first passage time to death conditioned on dying, or I could calculate the probability of division rather than death. And all those I can calculate by making this process. This process is also described by a matrix, but it's some funny type of matrix that's called A hat. Okay, now what happens if I have a histogram now? So again, I'm going to do that funny thing where I move the vertical axis to a horizontal histogram. But initially I have some number of widgets. So my histogram is peaked at some initial condition. As you wait, the histogram will broaden. What happens eventually? No. It goes to 0. These are absorbing boundaries. Right? So you keep losing things on both sides. Right? So once you start to have some number here, you keep losing stuff on both sides, and because you remove this, you never get it back. Okay? So I can calculate for this funny matrix, this histogram, what is the solution to this equation, by the way? dc dt is A times C, where A is not time dependent. Yeah, it's very simple. So C of t as a vector is e to the t a C of t equals 0 as a vector. And then right this way is because we have to do left multiplication. Yeah. It's different because we've removed what happens to cells after they divide. We don't care because I'm just interested in the first cell that hits the division. I don't care what happens to the second one, the third one, and so on. Okay, is that clear? So the usual trick for a first passage time problem is you pretend that the boundaries are absorbing. Those places where you want to check when it reaches first, you just make them absorbing boundaries. It's a trick. It's a mathematical trick. Okay. So what happens with histogram is eventually stuff starts to go to the left and the right. Now if I were to plot bin C1, bin C omega minus 1, right? The total probability of what went out on the right, right, would be what? It would be something like alpha omega minus 1 C omega minus 1 t integrated t equals 0 to infinity with some particular initial condition. Okay, so I'm saying a lot of different things now. What I've said is You have asked me, if I know a cell has this many widgets to start with, what is the chance it will divide? I tell you, give me a second. I take my original matrix, but I remove these terms. I then do matrix exponentiation. I then calculate, it's a lot of work, C1 of t and C omega minus 1 of t. And I know that the total number of cells that left on this side is the integral of C omega minus 1 t times the rate of leaving to the right. Okay? So this is the probability of division after infinite time has passed. Eventually all cells will either hit here or hit here. The chance that a cell avoids both boundaries forever goes to 0. Okay? P death is similar. It is gamma C1 t dt. Yes? Okay, but I'll come back to you in the discussion session. So this is the total flux that left on this side. That's the total flux that went out on that side. And so those are the total number of cells, or in this case, the fraction of cells that divided or died. Therefore, that's the probability of division or death. Okay? Now this looks, again, very, very complicated, right? But it's not. If you notice, let's just take this one. By the way, this plus this must equal 1. So you might as well just calculate one on the other, right? So this thing is gamma integral of C1 t dt, 0 infinity, right? So in general, you have some gamma times integral of C dt. The integral of C dt can be calculated directly from this, right? It's just 1 over a times C0. There's no 1 over a for matrix language, it's actually a inverse. Okay, so in fact, it's very nice that matrix integration and differentiation behaves just like your standard calculus integration differentiation, but when you take the matrix inverse instead of taking 1 over gamma or something like that, right? So I can actually, so this thing, integral of C dt, is actually simply a inverse C t equals 0. And this is the power of stochastic process framework, that I took a complicated question, I gave you a recipe to do it, that recipe reduces to matrix exponentiation, but you don't even have to do that. If the thing you're interested in is probability of division and death, you just take the inverse of this matrix, which in MATLAB takes no time at all, and you multiply it by the initial condition, seen as a vector. And then you take the first component of that vector. That's it. Okay, so that's what I wanted to stop with, right? I'm not giving you enough to become experts on first passage time problems. But I hope I've indicated to you that learning this kind of process, whereas earlier you would probably have gone and sat down and done some huge simulation, and if there's enough fluctuations, you might have to do 1 million replicates, and then eventually you don't know if you got the exact number or whatever it is. You reduce everything to a simple matrix inverse, and then you go home for lunch. Right? So there's a reason why people use this mathematical language, because by connecting with the power of linear algebra, calculus, and various other things, you don't necessarily have to do big computer simulations. Now remember, in the first half of the talk I said, I don't care if you can solve these problems. This doesn't amount to solving a problem. It's not a complicated Laplace transform way to solve a master equation. It is a realization that the problem can be reduced to a matrix inverse, which computers are good at. That's how I actually calculated those curves in that figure. Let me leave you with one last homework problem. This is not the mean first passage time. This is probability of division. What is the conditional average time it takes to hit this axis? That time will be something like you put a t in here. Right? It's the average time. So every cell that leaves at a certain time, you label it with the time it left, then you integrate over all the fluxes over all possible times. So having this t in here is the way to calculate the mean first passage time. Right? Except that not all cells leave to the right. So this is not a normalized distribution. So you have to divide this by the overall probability of division. And once you do that, you get the right answer. And I leave it as a homework for those of you who are more experts on this, to actually calculate for this very simple model not just the mean first passage time, but the distribution of first passage times. Because people have now started to measure these things experimentally. In single cells, they've started to measure the fluctuations in division times. You can see whether the simple model not only captures the averages, but also captures the fluctuation. If it doesn't, it's okay, but it'll be nice. It falls into some generic family. So I'll stop now. And I've been told to leave 15 minutes for questions, and there's a good reason for this, because I've jumped back and forth between like 100 different topics. So please do. I know a lot of you are saying, how does this connect to things I knew before, and how will it connect to what's in the school? So we're going to stop and have a discussion about that. Okay, thanks. Vijay. Yeah. Yeah. Actually, so technically you might be worried about it, but it's a finite matrix, and this is actually perfectly fine. It does. A hat. Thank you. Yeah, but this C is defined with a new process in mind, not with the original process. Yes. When I put the T in there, right? Yeah. It takes a little bit of time to realize this is what the mean first passage time is. What it means is, it's actually just this diagram, okay? Every time a cell hits this boundary, I calculate what time it left, and I write that in a table. I get a big table with a lot of different times. Now, the rate at which cells are hitting this boundary are actually these fluxes. So if I associate that flux with the time, then to calculate the average time, I just multiply that with the label, and then I average over all the label values, right? So, but I repeat the experiment many, many, many times, and I make a big list. On a computer, it's actually fairly straightforward. I keep on doing this. Every time it hits this, I record the time. And then I do this 1,000 times, and then I just take the average of it. So this itself is the distribution of first passage times. That's its mean. This is like an average. Probability distribution, P of x times x, integral over all x, is the average value of x. This is the probability of hitting the boundary at a certain time. This is the label at that time, so this is the average. You could do it that way, but in a computer you could do it that way, but here you'd have to be very careful. So this, that's why I'm saying that this is not a normalized distribution, because this is not a normalized probability. Not all things hit there first. So you have to divide this by C by P div. Sorry. This is not P div now. This is T div. Average is 1 over P div times this. Okay. Look, it is complicated. This is one of those things this is one of those things that you have to see many, many times to become comfortable with it. So don't worry if it's not all clear. More questions? Yes.
So the definition of C omega minus 1 is the number of cells having widget omega minus 1.
Correct.
So, first of all, I'm not very clear why the probability of division would be over all time, because with time, your C omega minus 1 would be changing, and your normalization constant would also change.
Yeah, this whole distribution actually drops down. The initial area of this distribution is 0. Okay? There's a flux going to the right, which is this quantity. There's a flux going to the left, which is this quantity. Eventually, the integral of this flux over all time plus the integral of this flux over all time must equal the original area, which was 1. And that's why P division plus P depth adds up.
Okay, just curious, what would be the normalization be of that probability, P div?
I've done 2 different things. There's no T here. These 2 equations are true. And the sum of those is 1. No constants. No constants. Right? If these are solutions to this equation, exactly, where the original C has 1, yes, and its total area is 1. Okay? So these 2 are true, and they both add up to 1 because things have to be conserved. Okay? Then I did a different thing. Then I said, these are just the probabilities. What about first pass systems? Turns out that this, the total probability of division is the probability of division for any time added up over all times. So when you think about it like that, you realize that this thing is the probability that you divide at any given time. Okay? And since now you have a probability of dividing at any given time, you average that multiplied by t to get the average probability. So this is the probability distribution of division times. Except it is not normalized because its integral is p div. You just divide it by p div to make it normalized. It is. You could do that. You could start your C initially as an entire distribution, or you could start with a fixed bin. You could do any number of games like that. No, it's not. It's only when you reach the t equals infinity limit that it looks like Markovian, but it's not. Individual cells, there's huge amount of memory and inertia. There's huge amount of inertia. That's why you get that transient. So in fact, you see the growth rate dropping slowly. Beautiful. In the experiments, you see it. Back.
Actually, probably my question was related to what you asked. So I was looking at this as a kind of problem in which you have separate timescales, in which you have a Phi timescale in which widgets get produced or die inside the cell. And then summing up all the cells which have a certain number of widgets, you get a random variable which is like live cells. And this summing up like spoils Markovianity, right?
Yeah, exactly. So
but okay, but then you answered his question. So I was asking about the Markovianity of this process, and if I can add another question is what happens if you have more widgets, so you have even more memory, introduce even more memory into your system?
So two things. So again, to read
in more kinds of widgets, I mean.
Right. Okay. So in the model, we assume that the widget process is a fundamental process which is Markovian by definition. The cell process is not. Well, in the sense it is when you plot cells in a state space where you know the number of widgets. But when we say the cell process, we mean, suppose I just told you how many cells there are now, and I say, how does that change over time? I can't calculate that. I need to know the distribution. Okay? And this comes down to a fundamental new way of thinking about things. Exponential growth can be explained by assuming that immediate post-division daughter cells reach a constant composition after some lag. And then that composition never changes. Exponential division and death can be explained by saying that the whole probability distribution of cells over composition space reaches a constant distribution. This is a very different way of thinking about this. This explains exponential growth and death. The previous one can only explain exponential growth. Okay? It's not to say cell growth and death is Markov. These rates simply correspond to leakage in the boundaries of some distribution in composition space. That's the first question. Sorry, forgot your second question.
Like, okay, this is a simple model. It's a very nice model of cell growth in which you assume that you have only one kind of widget. So you assume that you have a certain threshold above which the cell can divide.
So what happens if you have more?
More?
Yeah. So look, in terms of general philosophy of science, let's discuss how should we extend this model, what is missing, how much to take it seriously. I don't take this model seriously. I don't think cells are made up of fundamental widgets. I don't think that the division of a cell is determined by exactly when you hit a certain number. Okay? There are too many things in this model that are all coupled to each other because it's so simple. In this model, the widget number controls how long it takes to get to the division threshold. It also controls the level of noise. A real model will decouple both those things. In this model, unfortunately, as you increase the number of widgets, the noise level also goes down. You could imagine a more complicated model in which the noise level stayed flat, but some time scale increased. You know, all these things should be kept separately. So in this model, as you increase the number of widgets to even 50, it's pretty much the infinite widget limit. The infinite widget limit is very nice. The immediate, so as soon as the cell divides, you get exactly half, from omega you go to exactly omega over 2. And then that very tight distribution simply moves as the cell grows and hits omega again. And then it comes back here. So it's a very deterministic picture. So what happens? So now you're going to ask me more about widgets. So ask me, yeah. I haven't fully answered your question.
Okay, yeah, I wanted to ask, so what about having more kinds of widgets
Oh, yeah.
on which you put, so several
Well,
thresholds?
it's different kinds of things, right? So look, a cell is not a collection of...
So the omega changes the time scale somehow, but...
What is a real cell, right? So a real cell contains ribosomes, DNA, cell membrane, metabolites. Each of these things has a different scaling pattern with respect to cell size in some linear units, okay? They can interconvert. All these things then conspire to generate an immediate post-division daughter cell of a uniform composition, right? So that's precisely, you know, when I alluded to Sanjay's paper, how that happens is one of the open questions in this bacterial growth law world. And I think, you know, maybe we can discuss over lunch, but a cell has, even if you want to move from, you don't want to jump all the way to this crazy idea of a computational cell where I model this, the location of every molecule and agent-based and all that. You know, even if you don't do that, this model is not sufficient because it doesn't even capture the scaling of various different subsectors of the cell. Even small models that do that start to get very complicated. Lipids, metabolites, ribosomes, and those ratios. Many not even solvable, right? So it's a question of how we make any progress there. Luckily, we now have experiments where people have measured various proportions of lipids versus ribosomes versus, you know, actually last year's winter, or 2 years ago, winter school was all focused on, you know, a lot of this stuff. So in fact, some of those videos are still, yeah, so go back and look at some of the videos from previous years of this school to understand how that stuff works. Right? So I'll try and make a list of which ones of those to watch to answer that question. My goal here was not to talk about bacterial growth laws. The goal here was to use it as an example in order to show you that these kinds of calculations can be interesting and useful. Yeah. Yeah.
But it can be like volume or something, like when this...
Continuous.
Yeah.
So, good. I agree. Once again, I say the model is limited. No reason to expect a cell to be describable by integer quantities. And in any case, since we know that the number of widgets according to this thing is just a few, the model in some sense disproves itself. Because we know that the widget is no longer a fundamental unit. Right? So, there are lots of ways to extend it. Making continuous parameters is one. Yeah. By the way, continuous stochastic processes are pretty much the same as this, okay? It's not much more complicated. Instead of getting master equation, you get a partial differential equation. So that's not the stumbling block. It's not mathematics.
Yeah, maybe last time I could not pose the question in the right way. Now, may I ask, okay. If the way you derive that particular equation, the time evolution of CW, I followed it, but suppose I did not follow it and I'm just looking at the equation. My question is simply that it looks like a selection equation of CW, means C1, C2, C3 up to suppose C100, how they are getting selected. And if I interpret the terms like that, the second and third term comes from the cooperation of the closest variants. So I'm just asking that thing.
Your intuition for how to unpack an equation is a very personal thing. If it makes you more comfortable to think of it in that way, it's fine. For me, I'm more comfortable thinking of it as a vector in some space, and this matrix is rotating the vector. Right? All of us have arrived at knowledge of linear algebra through different pedagogical means. So you have to use the thing which you feel comfortable with. There's no standard way. This is again why it's it's good to have seen the same thing from even two of our different perspectives, and hopefully the contrast enriches your own. You pick and choose which ones of these ways works better for you. Yeah? Oh, you had a question which I didn't answer. Promised to answer it.
Thank you, sir. Yes. So I'm going to talk about the steady state, which I think it's easier to handle. So right there, suppose that you are not dividing CL into these each subcomponents, but you sum them. Okay? So you have here C0, and you have from C1 to CW minus 1.
That's this.
Exactly. And what you do is that let's picture it vertically. Okay, so you put it like here. And now you only have two components there. And this one is doing like this. And this one other is doing like this, right? And so I think at that point, it's clear that lambda is alpha minus gamma.
Right.
You know, so...
But that's not true for the transient, because using cells as your fundamental units, this is not a Markov process. So what you said is to ignore the fact that there are different W's and just pretend that it's all one CL, right? You won't get the right answer. Because in this model, there is more underlying dynamics. You require more information in order to calculate what's happening at the next time. That was the question Matteo asked. That's the question somebody asked in the back. It's not true that you can take a process at one level, coarse grain it, and necessarily get a very simple Markov description at the next level. In some cases you can, but it's almost always not true. Usually science goes the other way around. You assume it's some fundamental level at which you can describe it as Markov. You realize that's not true, and then you add a further level of detail and you go downwards. Okay? The level of coarse graining that's needed to address a particular problem is, in some sense, the fundamental question to ask when you model. There's no formula for it. Okay? Just experience, mistakes, comparison with experiments, rinse and repeat. Okay? And we can discuss the general philosophy of modeling again over lunch, but you're asking complicated questions, which I cannot answer except in specific cases. Yeah.
Right. So I'm going to say some words which I don't really understand. But when I came on the plane, okay, I was seeing this Einstein Nat Geo series, and he talked about this master clock, okay, which measures time. And now I completely don't understand that. I shouldn't be saying these words, but it seems like right now there is a master counter in the cells, you know, which is very debatable.
Oh, yeah. Good. Let's, now again, I'm not here to discuss bacterial growth laws, but since you asked and since there seems to be a lot of curiosity about it, there are big questions. Are cells constant adders? Are they constant ratio devices from beginning to end? Is there some other way in which they decide their cell size and division time? Turns out to be species dependent, but there is a general notion of an added portion of a cell. This is all stuff which you can find in the literature. Right? Here we have the simplest possible model. You have a counter, which roughly calculates the age of a cell, and the closer it is to the threshold, the closer it is to division, right? This is not how real cells work. And forget eukaryotic cells. I mean, we're just talking about bacterial cells. So you've asked things which are out there in the literature, and again, I'll direct you to some papers, read them, and then get back to me. I'll be here next week. We can chat. This was such a popular. One last question, I think. What I'm thinking. There is a notion that's good. So what's the other assumption hidden in this model is a notion of some sort of orthogonality. Right? So that is what I'm assuming that changing nutrients will change all three. But somehow changing antibiotics only changes a couple of them. The only way to start to do all this is to do a few experiments with different nutrient concentration, different nutrient richness and or quality, and different antibiotics. Just the standard type of growth law experiment, but now to calculate the rate of death also. Once we have that phenomenology, we can go for it.